Hello viewers, I am Dr. Mercy Anthony, Assistant Professor and Head, Department of Statistics from St. Joseph's College of Art and Science, Autonomous Cuddalore. I hope you will find this video informative, and by the end of this video, you will be able to understand the concepts of analysis of variance and design of experiments. So first we'll see what is the meaning of analysis of variance. Analysis of variance is a technique which is used to test equality of means when more than two populations are considered. This technique enables us to perform simultaneous tests and as such is considered to be an important tool of analysis in the hands of researchers. By using this technique, one can draw inferences about whether the samples have been drawn from populations having the same mean or not. Next, we'll see what are the basic principles of analysis of variance. We have to make two estimates of population variance, namely, one based on between sample variance and the other based on within sample variance. Then the two estimates of the population variance are compared with the F-test. So the F-test goes like F is equal to estimate of population variance based on between sample variance, which is divided by estimate of population variance based on within sample variance. In ANOVA, we are having two types of classification. The first one is one-way classification and the second one is two-way classification. First, we'll see what is one-way classification. As the name goes, it is one-way classification. So, when the observations are classified according to one factor, the classification is called one-way classification. For example, the yield of several plots of land may be classified according to one or more types of seeds. So here we are taking only one factor. So thus it comes under one-way classification. Next we'll see what is two-way classification. In two-way classification, data are classified according to two different factors. So for example, the yield of a crop in several plots of land may be classified according to different varieties of seed and different varieties of fertilizer. So here in this example, we are taking two factors. So thus this is the example of two-way classification. Next, we'll see what is design of experiments. So the definition of design of experiment goes like this. Design of experiments is a systematic, efficient method that enables us to study the relationship between multiple input variables and key output variables. So here the input variables is factors and the key output variables are the responses. So the next question is when to use design of experiments. So here I have listed four types. The first one is to determine whether a factor or a collection of factors has an effect on the response. The second one is to determine whether factors interact in their effect on the response. The third one is to model the behavior of the response as a function of the factors. And the last one is to optimize the response. Next, we'll see some of the important terminologies in design of experiments. The first one is the experiment. So what do you mean by experiment? An experiment is a device or a means of getting an answer to the problem under consideration. Experiment can be classified into two categories. The first one is absolute and the second one is comparative. The first one is absolute experiment, which means it consists in determining the absolute value of some characteristics like obtaining the average marks of a particular group of students or the finding of correlation from a bivariate population. So here I had given two examples for an absolute category. The second one is Comparative experiments. So comparative experiments are designed to compare the effect of two or more objects on some population characteristics. For example, comparison of different fertilizers, different varieties of crop, etc. The next terminology is treatment. So what we know about treatment is the various objects of comparison in a comparative experiment are termed as treatments. For example, in a field experimentation, different fertilizers of different varieties of crop or different methods of cultivations are known as treatments. Next is experimental unit. So as the name goes, unit, so which is known as the smallest value in any one cell or any one whole value. So the smallest division of the experimental material to which we apply the treatment and on which we make observation on the variable under study is known as experimental unit. So for example, here I've given you the example. The field experiment, an example of field experiment, a plot of land is the experimental unit. In other experiments, unit may be a patient in a hospital, a batch of seed. So it is also known as sample. Next, we'll see what do we mean by yield. The measurement of the variable under study on different experimental units are termed as yields. Next, we'll see what we know about experimental error. The variation in responses caused by the irrelevant factors is termed as experimental error. The experimental error may arise due to three factors. The first one is the inherent variability in the experimental material to which the treatments are applied. The second one is the lack of uniformity in the methodology of conducting the experiment. And the third one is lack of representatives of the samples to the population under study. So these are the three experimental error which can be caused. So I have taken one example here. Let us consider one agricultural experiment. And in suppose we are taking a large homogeneous field which is divided into different plots of equal shape and size with different treatments which is applied to these plots. The yields from these plots are observed. Even if the same treatment is used on all the plots, the yields would still vary due to difference in the soil fertility. Such variation from plot to plot, which is due to random or chance, which is beyond human control, is known as experimental error. Next, we'll see what are the basic principles of experimental design. So in here, the three basic principles of experimental design are listed. The first one is replication. The second one is randomization. And the third one is local control. So these are the basic principles of experimental design. So we'll see the first one, the replication. Replication means the repetition of the treatments under investigation. So an experimental resorts to the replication in order to average out the influence of the chance factors on different experimental units. Thus, the repetition of treatments results in more reliable estimates than is possible with the single observation. So as the name goes, repetition. So repetition or replication serves to reduce the experimental error and thus enables us to obtain more precise estimates of the treatment effects. Next is randomization. Randomization here means is the process of allocation of treatments to experimental units at random so that each treatment gets an equal chance. Randomization provides the logic basis to draw the inferences by the use of statistical theory based on probability theory. It ensures that different treatments by the repetition of the experiment on the average are subject to equal environmental effect. Randomization eliminates bias in any form. It equalizes even factors of variation over which we have no control. Next, we'll see what is local control. Local control means the control of all factors except the ones about which we are investigating. The process of reducing the experimental error by dividing the relativity heterogeneous experimental area into homogeneous blocks is known as local control. So here I had given you one example, like if we are taking one agricultural field for an experiment, which is heterogeneous and different treatments are allotted to various units at random over the entire field, the soil heterogeneity will also enter the uncontrolled factors and thus which will result to increase in the experimental error. In short, it may be mentioned that while randomization is a method of eliminating a systematic error in allocation, thereby leaving only random error component of variation, the other two, namely replication and local control, try to keep this random error as low as possible. So in general, to have precise, valid and accurate result, we adopt the basic principles like the three basic principles: randomization, replication and local control. Thank you for your patience listening and I hope this video will be useful for you. Thank you.